Hello, everyone. I will try to reclaim 1 or 2 minutes of my delayed start. I'm going to switch it up a little bit and talk about diversity and embracing diversity, specifically within the context of MPS. I'm still out of breath from running from the restroom. So I'll take a high-level sort of look at obesity and its impact from a global standpoint, as that is our core expertise, and then dive a little bit more into what that knowledge gap translates to, what we do at Oberthala Sciences, and so the tissue model that we make, the models that we provide to help address this gap, recognizing the value that this may present from a diversity standpoint, and then looking at ways, different institutions that support the cause, that are recognizing the value of diversity, that are recognizing the potential impact there from a global standpoint. So I'm going to talk about, I said I'll start off with obesity, and looking at this as our core expertise. When you look at this map, this is from 2020, the CDC reports. If you look at Louisiana, this is where we are today. We're ranked 4th, unfortunately. This is, one of the stats that we're not proud of. All the delicious food that we have, being ranked 4th in the nation with respect to obesity. And when we look across the board, though, obesity is impacting 20% of adolescents, 42% of adults, and it's an epidemic. And it's costing us on average, about $100 billion from an annual economic perspective clinically. And that easily triples when we account for some of the other metabolic diseases that I'll talk about in just a moment, like type 2 diabetes. And so now, because of these communicable diseases and because of COVID-19, for example, SARS-CoV-2 as a virus, we see that obesity is an independent risk factor for COVID-19 associated hospitalization. And we're starting to have more of an appreciation for the role of adipose and obesity itself. And so I don't need to tell everyone here that there's a need for platforms that provide earlier indications of the human response. Neither do I have to tell you the importance of those platforms. But what I will talk about is, within our specific context of obesity, the 11% attrition rate. And that this not only is being reflected in obesity itself, but also the increased risk for the development of some of these associated diseases. And cancer is one of those areas by which is now being more appreciated as a metabolic disease. And so we're looking at, yes, we all know that an animal model is not a human, but what are the other opportunities that we have to address this problem? So we have to look at the real problem. What are the compounding factors here? It's not just that we have a poor attrition rate. What is contributing to that? Not only is a mouse not a human, but what are the other things that we're missing at the clinical data? So representation of patients of varying ages, genders, in some studies, body mass indices, for which compounds are meant to treat, right? Do we have representation of morbidly obese individuals who are over the age of 50 who may be getting, essentially types of the therapies that are not presenting with significantly positive rates in terms of their success? So can we look at the historical context for this? And I'm building the case and the rationale for looking at incorporating diversity within a specific context. But if we look historically, we're taught at some of the earliest stages within our early biomedical engineering classes or even biology classes that structure and function go hand in hand. We look at this from the standpoint of, if there's a function of an organ, what is the job of that organ? But that's directly impacted by what the organ is made of, right? What is its composition fundamentally? And this is still being tested within different organ systems today, even though it's a very foundational principle that arises out of observation within the context of overall system and complexity. And so we look at the cellular composition. We're looking at the different elements that contribute to the functional outputs because of the functional relationship between the cellular components and the structure of that tissue. So we have demonstrated in our laboratory and some of our collaborators' labs that mesenchymal stem cells, the population that we use and have tested from adipose tissue and bone marrow, that those cells retain some of the key features of the host that those cells are derived from. And so we looked at things that are representative of differences on the basis of anatomical location, for example. So with adipose, subcutaneous tissue is not the same as visceral tissue in the way of some of its functional elements and contribution to the overall metabolic homeostasis. And that's partially due to the difference in structure, which is due to the difference in the stromal compartments. We also see differences across different anatomical locations within racial ethnic groups. So within, for example, individuals of African ancestry, comparing their breast tissue, whether it's normal tissue or in the context of breast cancer, to other populations, we see differences in density, which correlates again to the stromal composition. We also see differences in gender, and these studies were done within murine populations. But we look at the clinical, the historical points that were provided, and we're able to generate these relationships based on what we observe within a tissue level, dissociating that tissue, getting those representative populations, and then plugging them into systems like what we provide. Oops. So essentially what we're doing is we're learning from the literature, we're taking some of the historical data, we're looking at the structure-function relationship, and then we're saying, okay, can we look at that, build on it, apply it to current day technology and advancements that we have? Can we essentially address this or fill this black box of functionality? It's essentially described as such when we say we have a microphysiological system that represents the functional output of organ X. Quite often what we get is, well, I don't quite understand how that works, but I'm going to trust you that what your system is doing, it actually does provide that. And we're asking, can we fill this knowledge gap in treating the diseases using this structure-function relationship? And so at Obatala and all of the phenomenal technologies that have been represented here, we are all, of course, taking it back to the human. We're looking at, within our specific context, knowing that there are differences with respect to the cellular composition. Let's clearly define these cells. Let's clearly define all of the elements that go into these systems, these model systems. And in our particular model system, we're looking at making sure that the cells that we use, the cells that we start off with, that we are well characterizing them. So we start off with things such as the proliferation rate, the colony forming units, the immunophenotype, the adipogenic and osteogenic differentiation. We incorporate all of those elements with the demographics of the patient. So we're providing much more information. We're looking at starting with an earlier passage of a population of cells that we know does perform consistently. And so how can we use those starting points to generate these models that will represent the patient of interest or the demographic of interest, knowing that we have all of this data? And so I did talk about some of these, the demographics that we start off with, flow cytometry, looking at these cell surface marker profiles of these cells. And the other element that we use that we're incorporating into our tissue model is a human-derived hydrogel. So we are manufacturing these hydrogels that are naturally derived that can replace or serve as an alternative to mouse-derived materials, even to some of the synthetic protein-derived materials or hydrogels. And so again, we're looking at, okay, we need to make sure that all of the parameters that we're testing in terms of characterizing these hydrogels, that they are yielding consistent output, that they are yielding high quality in terms of the type of response that we're looking for. So supporting and obviously being biocompatible with the stromal stem cells, and it extends outside of the cells that we use, but also in the context of iPSCs and other cell types. Our hydrogel has been used for supporting those applications. I mentioned looking at the batch-to-batch consistency and performance, and for us that's as simple as pooling and looking at the differences with respect to the minimal lots that have to be pooled to yield those similar outcomes. And of course, something that is adjustable in terms of tissue stiffness. We've been studying the viscoelastic properties and characterizing those with respect to the hydrogels that we manufacture, including this hydrogel here, Obagel. So start off with the quality cells, for which we know these cells are retaining some of the properties of the patients that we derive them from. Combine that with high quality media and hydrogels, and what we get is a system that could be used, whether it's a service that we provide within a multi-well format, whether that's in the context of a perfusion bioreactor or in our customers' own laboratories. And some of the other applications extend beyond. So what we do in our lab, we have the multi-well system, which is a static system, 96-well format, or down to smaller multi-well formats. But we also have had the hydrogel in the cells used in the context of organoid development and supporting PDX studies. And so the various applications of this particular hydrogel in general, it's far and wide, because now we're looking at a human-derived material that is manufactured as a research-grade product, for which the goal is to generate a clinical-grade product in terms of manufacturing. So we can get into the areas of personalized medicine, cancer therapeutic screening, when it comes to patient stratification, those types of applications. And again, taking it back to the point of incorporating elements of demographics of interest. And so when we talk about representing patients of varying demographics, and again looking at the cellular composition as that contribution, the way that we do that is by starting with the stromal vascular compartment. That is a heterogeneous population of cells, which not only includes the adipose derived stromal stem cell, but also the immune cells. There are some B and T cells that are native to adipose tissue that have been well characterized within the literature. The vascular elements, which are presented, are contributed by the vascular smooth muscle cells and the endothelial progenitors. And we have demonstrated that we can measure the functional outputs with respect to glucose uptake, lipolysis, those typical markers of adipose functionality, in combination with maintaining some of the subpopulations that are present within adipose that contribute to these differences. And so I mentioned a little bit earlier that we can support organoids. This happens when I'm in my office, the same thing. It's only because I started talking. And so I mentioned earlier that we have supported and can support organoids, spheroid culture, for cell encapsulation, for multi-well format, static or perfusion bioreactors, or even in the case of PDX models in terms of the flexibility of this human-derived gel. But this is just one example in which we have spheroid culture, and there's essentially lipophilic dyes indicating the adipogenic functionality of the cells here. And we basically demonstrated that increasing the percentage of the hydrogel does increase the density and the size, the diameter of these spheroids. We're able to recapitulate the subcutaneous adipose tissue. We looked at cells derived from subcutaneous adipose depots. And so, of course, we have to keep track of that information to generate these types of models. But in this particular instance, we're able to establish a not a crown-like cell, but a clear indicator of a, oops, let me go back. There we go, of a unilocular cell. Essentially, the white adipose, the lipid droplets get very large. We get unilocular type phenotypes compared to multilocular adipocytes when we are inducing more of a browning or a beige-like thermogenic response, which is an upregulation of uncoupling protein 1 and activation of mitochondrial activity, which can also be measured by oxygen consumption rates. And we also get corresponding differences with respect to the adipocyte area and the nuclei within the 3D constructs. And I won't even mention the 2D because everyone here knows some of the inadequacies within certain contexts. I won't say all with respect to 2D, but in the context of brown adipose tissue, that also has been well documented within the literature that a three-dimensional structure is superior in terms of its functionality. And one of the other advantages of having a human blood-derived hydrogel at market is that it robustly supports vascularization. I mentioned that earlier, but we were essentially demonstrating this by culturing cells, encapsulating endothelial progenitors, allowing them to form these very nice networks that we can get consistently within each of our structures with our models. And so looking at functionality, adipokine secretion, adiponectin and leptin. Looking at glycerol secretion, we get similar functional outputs to excised adipose tissue. Similar with respect to lipolysis, So glycerol secretion in response to isoproterenol, metformin, compound C, so classical AMPK targeting compounds, in which when we induce this robust response to isoproterenol, we get something that is not dampened by the presence of metformin and compound C, which has also been demonstrated in the literature compared to a 2D system. You don't get that type of a difference in the inability to block its activities due to its activity on beta-adrenergic receptors within cells. And so one of the other things that is really a hallmark about establishing these types of constructs is that you can then develop, I mentioned PDX support, but we have looked at in the context of xenograft models, so establishing a human subcutaneous adipose depot within a mouse, within C57 Black 6 animals. The hydrogel supports not only the migration of the host cells into and support of an adipogenic response, but also a very robust depot, a humanized depot within the animal. And so looking at institutions, NCATS is one of them, obviously, for supporting this meeting, this summit, which is the first of an international, which I'm very excited for all of us to be here and present such outstanding technologies. But there are also other branches, the National Institute on Aging, for example, which has supported our work and is continuing to support the inclusion and diversity with respect to systems like ours. And so NIA is sponsoring, for example, and has sponsored a webinar. It was recorded, it is old, so I said here's a link. But this webinar was essentially discussing the need for diversity and how diversity impacts science. So we're promoting diversity in research with respect to the types of tissue models that we're bringing forward, and also highlighting the value of incorporating those different demographics. But there are other ways to promote diversity in research by supporting scientists, by supporting opportunities for those new budding scientists to matriculate through these strong programs and get exposure to the types of technologies that we all are bringing forward. And bio is another one. I'm about to talk about a panel that they are sponsoring as well. NSF has their Office of Equity and Civil Rights, which is recognizing not only is there a need for representation, but also protection of individuals who feel that they don't have a voice, essentially. And so, what we started out with was looking at structure, function, the historical relationship, but then taking it down at the cellular level and how we're using that to bring diverse models to the market with respect to metabolic research within the context of obesity and type 2 diabetes. With respect to the standards, I didn't talk about that as much. Besides the context of us looking at standards with manufacturing the materials that we're making. And so we highlight that that is also just equally as important when we're designing these systems to incorporate these elements that contribute to consistency and performance. So a big thanks to our team members. I cannot thank David and the team enough for their support of bringing everything together for the summit, and also the NA3RC, all of the phenomenal researchers, phenomenal individuals who are working to collaboratively bring forward technologies that can reduce and refine the number of animals that are being tested on, as well as our partners and sponsors. And so I mentioned BIO. This panel on June 14th will take place. It's fostering diversity and bioequity through novel financing and training initiatives in the aging. And it will be established, or at least a composition of individuals who come from industry, individuals who come from the venture capital side of supporting technologies like ours, and from the NIA. And so the other piece of this that we have as motivation is not only having this larger impact, this broader impact from a diversity standpoint in research, but also the funding that's there to support the inclusion of more diverse samples and more diverse technologies, bringing them forward to the world. It is growing. So thank you, everyone. I would be happy to address any questions.